have 15 minutes of question and answers. Please switch off your phone or silent your phone for the further time. Thank you.
Thank you very much, Namaskara. Uh, it's a great honor to be here, and I think it's with great trepidation that I'm talking to you. First of all, I apologize that I'm giving this talk in English. I would have preferred to give it in Kannada. If I give it in English, only you won't understand. But if I give it in Kannada, I won't understand and you won't understand. So it is better that I talk in English. But Dr. Kolagalu Sharma is here. Afterwards, he'll give a summary in Kannada. The second challenge I have is that I have to keep the talk in a manner where the little kids in the front row and others in the last row, all of them understand it. And again, it is not a problem at your end, it's a problem of communication at my end. So I hope I can manage that also reasonably well. Now, I'll try to keep the talk brief so that there's enough time for questions and interaction. And thank you again for having me here. This is such a wonderful uh, program which you have. I've heard details of what all you've been doing from Dr. Kolagalu Sharma, and it's absolutely fantastic. And it's very fantastic because today's world, uh, as a result of how our brain has developed, humans have become not only those who have managed to survive on this planet, but we have become the principal agents of change on the planet. Today, we actually rule the planet, and no other animal or plant or any organism has done this in this manner. Every other life form has managed to survive on this planet, perhaps even dominate the planet. But the capacity for change which we have on this planet and what change we have done, no other organism has. How did that capacity of change come about? How did this brain develop? And what is our future on this planet? And those are matters which I'll try and address. Very briefly, these capacity of changes came about because of a combination of our body and our rest of our body and our brain working together on the environment. Because we can hold our thumb and forefinger together, we can grasp objects and we can make tools and we can shape the world around us. This is a very major change which we can do, which not any other animal can do in this scale. The second is our ability to vocalize sounds and through that create language, an interaction between our vocal cords and our brain. Liberally, this chair here also. Our interaction between our vocal cords and our brain means that we have created a software in the brain which allows us to communicate in a manner, and the brain itself has other hardware which allows the creation of new kinds of software which allows us to imagine, create abstractions, and do things in a manner again which no other brain can. So the software capabilities, apart from our making tools, has completely changed the world. And of course, as a combination of these two, there is what is called cultural transmission, cultural evolution. We have so rapidly learned things, and on basis of what we have learned, we've built new kinds of ideas, and this cultural change has gone much faster than the genetic change. So the result is we have become, as philosopher Dan Dennett said, we have become those who wield the paintbrush on the canvas of the earth. And the future of the earth depends on us. Today, the hazards which are there because of climate change, loss of biodiversity, environmental degradation, all of that has been caused by us substantially. And therefore, the responsibility for reversing it lies on us because it's not just our survival which is at stake, but the survival of the planet. So our job today is to try to understand how the brain, which is at the center of all this, which has done so many wonderful things, as well as so many complicated things on this planet, how is it made? Now, if you look at the brain, it is easy to say, oh, it is very complex. It has got billions of cells, nerve cells, which are its units, and it has got trillions of connections of these units, and it is very difficult, it's impossible to understand. How can we ever understand it? So that is one way of looking at it. It is true, the brain is very complex and very difficult to decipher at all its levels. But we have made enormous progress in understanding its components, how those components connect with each other, and in some ways, how they function together. But it is not just that the brain which is complex. If you look at a simple bacterial cell, and this is about 12,000 times smaller than a nerve cell. This bacterial cell, which you can't see, bacteria are all around us. This bacterial cell gets inputs of temperature, senses the sugars and carbon sources around, senses other chemicals around, and it takes decisions on what to do, whether to move in one direction or stop or move in another direction. We have understood many aspects of this bacterial cell, but we have not understood all aspects in a manner where we can predict everything the bacterial cell can do. So even in a simple small cell, despite our enormous understanding, much remains to be understood. And therefore the question again arises, if we know so little about this one small cell, how can we understand billions of cells put together and how they function? So for example, in a bacterial cell, it can sense the sugar, which is shown on your left side. There are receptors of the cell which will sense the sugar or other kinds of carbon sources. And it will change the tail, the flagella. There are many, many flagella here. It will rotate them in a manner so that the bacteria moves in one direction or the other. So bacteria can sense the environment. And we've learned a lot about how single kinds of sensory inputs, sugars or carbon sources or temperature, and how the bacteria responds to it. But we know very little about how when multiple inputs come together at the same time, how the bacteria will respond in a predictive manner is something which we still don't fully understand. Bacteria are thought to be unicellular, one single cell. And we and our brain, have got many, many billions of cells. But even this single cellular organism talks to others. So bacteria, oh, so even a single cellular organism like bacteria communicates with others. So these bacterial cells here will put out some chemicals which will talk to other bacterial cells and they will change the properties of the neighbors. It is as if all of us are bacterial cells and a small number of them put out a signal and everyone behaves the same way in response to the signal. Right? So that bacteria can do. And this has been very nicely put together by a famous researcher at Stanford, Bonnie Basler. You must look up her TED Talks. She's absolutely terrific in conveying how our understanding of bacteria and how they behave as communities has changed. So, this is an example of how by studying biology deeply you get an understanding. So, how can we get an understanding of something as complex as the brain, which has millions of neurons, billions of neurons? Now, one way is to start looking at it bottom up, look at the units and how they function and how they're put together. That will give you some amount of understanding. If you want to understand how a huge, big architectural marvel has come across, a big bridge or a temple, you can say, one route is to say what goes into it, what are the components, what are the bricks, what are the steel girders, how are they put together, and slowly bottom up try to piece together how you can construct this bridge. That's one way of looking at it. So there were two famous scientists, Hodgkin and Huxley. They took this giant squid, which is a sea animal, and this squid has got what is called a giant neuron, a very long neuron, and they started looking at the electrical properties of this neuron. Now, a neuron is a nerve cell, and we have billions of nerve cells. And nerve cells communicate from one nerve cell to another substantially by signals which go from one end to another of the nerve cell. Those are electrical signals. And then each nerve cell communicates to the next nerve cell by a chemical signal. Broadly, this is the way it happens. So they asked, how does the electrical signal from one end of a nerve cell to another end of the nerve cell, the action potential as it's called, how does it propagate? So they looked at the physiology of that. Then they did something rather interesting. They could record and look at the signals going up and down. They also squeezed out from inside the nerve cell its contents and replaced it with different compositions of CLI. And through that, they again recorded. After doing that, they also recorded again. And because of these experiments, they predicted theoretically how this physiological output changed. They predicted what is the composition of channels, as they're called, which conduct specific ions, sodium, potassium, calcium ions, what the composition of these and the way they function. So this is a good example of how looking at the output of the neuron, its electrical activity, you could predict what the chemical components of a neuron. This is a beautiful piece of work. Now, this is a beautiful piece of work, and there are many good accounts of this. But this is one neuron. There are, for example, one kind of a neuron called the Purkinje neurons, and that's shown on the left here. It is shown as an anatomical drawing on the left and a high resolution microscopy on the right of a real set of neurons. This anatomical drawing was made by Santiago Ramon y Cajal in the early 20th century. And there are 50 million such complicated neurons. So the squid axon was simple and you could understand it. Here is the Purkinje cell, which is much more complicated, and you can sort of begin to understand it, but there are 50 million cells. And they are just a very small part of the numbers of cells in the neuron. Now, not only are they such a large number of cells, imagine if each cell is connected to thousands, 10,000s of other cells. Then you not only have to understand each cell, 50 million, hundreds of millions, but how they're connected and how they function. So this is again a picture, a drawing from Ramon y Cajal showing different kinds of nerve cells connected to each other. And this he did with a simple microscope and a special kind of staining which allowed him to see individual cells. So he could draw the kinds of connection. Today I'll come to a little later. We can look at connection at a much finer scale and also on a larger scale, larger number of neurons. So for many of these kinds of studies, we have learned about how the nerve cell is. The unit of functioning in our brain and in all brains is thought to be the nerve cell. It has got one part here where all the DNA, the genetic material resides, and also many other aspects of function which allows this cell to function. The cell has got this area which is called the cell body, and it's got a long axon, and that connects to another nerve cell through what are called synaptic terminals. Also, there are other kinds of inputs which come into this nerve cell. Just as this nerve cell communicates to that nerve cell, this nerve cell receives inputs from others. And there can be many thousands of inputs coming in and many thousands of inputs going out. So electrical signals typically go from here to here, and then there are chemical signals which go across, which causes another electrical signal. So that's the way it works. And this is a very important finding. These findings are all, they took many decades of work. Now, you can ask, Since these nerve cells connect to each other, there must be some simple circuits. Instead of looking at huge computers, if you look at a very simple small computer, and which has a small number of connections, can we at least understand that at a high level, right? So well, we may not easily understand the entire brain, but surely we can understand small circuits where about 5 or 10 or 15 nerve cells, neurons are connected to each other. That was what Eve Marder at Brandeis University, she's a very famous professor, she has been studying for many years the lobster stomatogastric neurons. What the lobster does, it keeps pushing food in a rhythmic manner into its mouth, filtering it and taking the small particles of food and throwing out the water again. So this is the way it feels. So this rhythmic circuit, what Eve Marder and her colleagues discovered, is caused by a small set of nerve cells called a central pattern generator. And she studied the central pattern generator, the nerve cells involved in the lobster somatogastric circuit. Now, this seemed such a task which was a minute. It was very, she took decades, students, postdoctoral fellows, and she took decades. There are a small number of nerve cells. You need to look at the circuit dynamics, and you need to look how changes take place. If you kill or ablate one nerve cell, how does the others compensate, and how does it stay stable because even despite changes. So during aging of the lobster, for example, the anatomy of this circuit changes hugely. These kinds of ramifications which you see here and here, they change a lot. Yet the lobster eats the same way. How is it? As the lobster grows, the behavior remains constant even if the circuit changes. So these are the kinds of things you study. And the short answer is that we've got amazing insights onto how, from this study and others, how small circuits work, but we yet don't know in a predictable manner how these small circuits respond to multiple inputs. We know a lot. We can predict a lot in some ways, but the totality still evades us, even though that is getting better and better at this. Now, I've told you about why the brain is very complex and how this bottom up approach is paying enormous dividends and how still complexity remains. But there are some major simplifying features of the brain when we study it. Even though there are different parts of the brain, they are more or less specialized. There's overlap, but they're specialized to do different kinds of functions. Some parts deal with visual information more, others with motor information, yet others process, sound, yet others process smell. So even though the units of the circuit are identical and their connections are based on similar principles, there are many different kinds of connections. The regional specialization allows the brain to be understood because you can study different regions and see how they function. This is like saying that there is one part of a city where there are sports complexes. They also use bricks and cement to build those complexes. There is a housing complex that also uses bricks and cement, but they use it in a different way. And they talk to each other, right? All the sports takes place in the playing fields, all the takes place in one place, the office is in another place, and there's communication. So it is trying to understand, despite the same building blocks being made, how these different components interact with each other and function. So once you know that there are different parts and they function in a different way, one can start studying those and seeing how it functions. And again, people have done a lot of work. Now, I told you that the building blocks of the brain across this whole region of the brain are similar. But actually, the principles of making this building block are similar across all parts of our body, not just for the brain, right? In all parts of the body. The body develops, as in all organisms, all multicellular organisms, from a fertilized egg That egg divides into 2 cells, 4 cells, 8 cells, and so on. And as the division takes place, there is different parts of these cells, different numbers of these cells, different parts. They become specialized to do different kinds of functions. Some become arms, some become legs, some become liver, some become heart. The rules for specifying different parts of this use the same building blocks again. And these building blocks are used across all organisms in the planet. That is, the building blocks which are used to make us are the same building blocks which are used to make a worm, a fish, and a plant. So, one of the greatest achievements of modern biology is to understand in a unified manner how organisms are built by the same kinds of building blocks. So whether you are a fly or a human, the same principles apply. What are those principles? One principle is I told you cells keep dividing. One way is as they divide, the mother cell gives rise to two daughter cells. Part of the composition of the mother cell goes to one daughter cell, and the other part goes to the other daughter cell. If they are asymmetrically distributed, these parts, then you create differences between the two daughter cells. Then if the two daughter cells again do that, you get more and more differences, right? So one way of creating differences between cells is by lineage, by inheritance. You give something to this cell, and you give something else to another cell. Another way is to say that this cell actually sends out a signal. It sends out a signal which makes all these other cells different. Near the source of the signal is very strong, and when a strong signal reaches one cell, it responds in one way and changes. And as you go further and further away, the signal is weak, and at a weak signal, the cell develops differently. So all cells are similar, but because of the environment, they behave differently. Right? So one is lineage, parentage determines what happens. The second is neighborhood. Neighborhood determines what happens. The third is the signal doesn't travel very long distances. It just travels to the next contact. It is immediate neighborhood which influences. So these are the broad kinds of mechanisms by which cells change and become different from one another. And slowly by using these rules again and again and again, I was very happy to see the work modeling of clay which the school children have been doing in the next room. They take the same clay, but by applying the same principles again and again, more here, less here, in a different way there, you can make features out of the clay which otherwise were uniform. Those rules about getting features out of a sheet of cells, those molecules are conserved across all of evolution. And so you can understand how a fly is made, a worm is made, and a human is made because they use the same kinds of upstream principles. At the bottom end, whether you are a human or not, or a fly or not, there are different reasons for that, and we'll come to that. But the principles are same, and that's illustrated. If you look at the expression pattern, genes make RNA, which makes protein, and proteins are there in different parts of our body. If you look at the expression pattern of proteins that's shown on the top in a fruit fly embryo, and you look at the expression pattern in a mouse embryo or a human embryo, the same kinds of proteins, the same kinds of genes are expressed in a similar pattern in the mouse as in the human. I won't go into the details of this, we can talk about it if anyone's interested, but these players are common again and again. Okay, now let us ask then, with these rules and principles, how can we understand how the brain is made? What I'm going to do is I'm going to, yeah, I've inserted a blank slide because I want to tell you a little bit now, using these rules, how the brain is made before I show you a slide. The slide might be a little too complicated. So Just as you start from a fertilized egg and you divide and make a body, the brain also starts from a small number of cells. Typically, these cells are stem cells. And what is a stem cell? A stem cell is a cell which, like many other dividing cells, gives rise to two daughter cells. But it does something different. One daughter cell is also a stem cell, and the other daughter cell is not a stem cell. Then the stem cell divides again, gives rise to another stem cell and another daughter cell. So these sets of other daughter cells which form, they then do something called differentiation. They become nerve cells. So a stem cell gives rise to a stem cell and a nerve cell. Stem cell and a nerve cell. So you can get many nerve cells. Sometimes you can also say you have situations where a stem cell makes two stem cells, four stem cells, eight stem cells, amplifies very rapidly, and then it starts making a stem cell and a daughter cell and making nerves. Once you make a nerve cell, that nerve cell now becomes into a neuron. I mean, that nerve cell progenitor, precursor, undifferentiated nerve cell, I should say, becomes a neuron. It sends out its processes and connects to other nerves. So, where this process happens is important. If it happens in the head of the animal, these nerve cells contribute to the brain. If it happens in the spinal cord region, it contributes to the spinal cord. If it happens in the limbs, it will contribute to the motor neurons and other neurons of the limb and so on. So, two things when you make neurons, where does it happen? The second is when does it happen? The timing, right? So there are rules again, just as there are rules about what kinds of nerve cells you will make depending on where the nerve cell is born. When the nerve cell is born in a particular space, decides also what it will make. So if it's an early neuron, first stem cell dividing, it will make one kind of neuron. If it is late, it will make another kind. So spatial and temporal regulation results in the diversity of nerve cells. And the spatial diversity is not just head, thorax, abdomen, but in the head and thorax along these axes and dorsal. So by using the same building tools again and again, but doing it differently in different locations, just as you use the same components, and there are rules. If you are got a map to make a stadium, you don't accidentally. Sometimes you might accidentally make an apartment building, but that is because the map has got exchanged. That also can happen because of defects. But when you have a map to make a stadium, you use the same building tools and make a stadium, and where you have to make an apartment building, you do that. So we'll come to later if you have time about what the map is. That's it. And that's what is shown here in this slide. Oh, sorry, I'll try to bring this down a bit. Yeah, so here is a stem cell marked NB that gives rise to one stem cell and two neurons and so on. And these neurons in red and green, they go to different locations and then they form contacts. So there is spatial patterning depending on where you are and temporal patterning depending on where you're born. Now, by doing this for different parts of the brain thoroughly and understanding it, in simpler, so-called simpler organisms, such as a fruit fly, you can map out in great detail every single stem cell which gives rise to neurons and how the neurons are connected. There are tens of thousands of neurons, not billions like us, but on those tens of thousands, you can do both detailed light microscopy and electron microscopy and get maps. You can map every single neuron, and thousands of young students look at the electron microscopy sections and examine it, and now it's also done through artificial intelligence. You can get detailed maps. This is shown before the annotation and after the annotation of this thing. So we know the connectivity and the function and the behavior and the rules quite a lot about this fly brain enormously. Still, our understanding of how the brain relates to behavior is an exciting area which we're trying to do, but is not yet fully happening. So, can these general principles, some of them apply to the human brain, others we don't know whether they do. One general principle which applies is that we know that the kinds of molecular mechanisms to make a brain or any other part of the body are conserved, and all organisms can make it. So that is shown over here. But are the rules which apply to simple circuits apply to multiple kinds of complex circuits? These complex circuits get inputs not only from the outside and respond like the simple circuits do, but they get inputs from simple circuits and analyze that, which analyze, so there's a higher and higher level of meta information which goes and feedback loops, so it's very, very complicated. So do the same principles apply? Is it worthwhile knowing the connectivity of the human brain like it has been useful in knowing the fly brain? Is it worthwhile trying to do that for the mouse brain before we do to the human brain? These are all open questions. Now, let me end by trying to wind down by asking what are the special properties or unusual properties of the human brain? Why are we able to, when we are listening to a talk, our mind wanders off to our family or to our work or something else because of something which you see here. That's why I put blank slide. You're not distracted. Or sometimes you concentrate, sometimes you wander, right? Do other animals do the same thing, right? When I show you a picture of rodents and monkeys, you think of Mysore Zoo, oh, I saw a monkey there, or this happened, or that happened, and so on and so forth. Does your mind wander? If a dog were watching this, would it also do the same kind of thing, or is it all completely, unclear of what a dog is thinking? So these are the kinds of things which that we know we can ask humans what they're thinking, and they will respond. So we can get an idea about what humans are doing. Now, if you look at human brains, one view is that the human brain, one view was that the human brain is very big, and therefore it can do all the things which we can do. And so a fly brain is small. But it's important to keep in mind that a fly does its small things much better than we can ever do. A fly can fly, we cannot fly, right? But a fly cannot do many of the things we can do. So small brains are very highly specialized and they are no less evolved. Our brain is unusual that we can do multiple things in a manner, integrate all of that, and as I said, change the world around us. Now let us look at brain sizes and neuron sizes between some very large rodents. Capybara is one large rodent. Its brain is very heavy. It's got 76 grams of brain, and it has something like 1,600 million neurons, right? But if you look at a capuchin monkey here, okay, which is a primate like us, its brain is only 52 grams, much less than this, but it has 3,000, close to 4,000 million neurons. So somehow primate brains seem to have many more nerve cells than non-primate brains. So that is one lesson. The rest of the weight of this brain is because of non-neuronal cells, what are called glial cells and other kinds of cells. So primate brains have a large number of neurons. For example, at 16 grams, the owl monkey has about 1,000 million, 500 million neurons. But at 18 grams, the agouti rodent has only close to 900 neurons. Right? So this is some very important. This is work done by a Brazilian scientist who works in the US now. Her name is Susana Herculana Orzal, and she has found a very nice way of counting the numbers of neurons. Because earlier people used to just look at brain weight and brain size and not nerve cell number. And she's done that. When she does that now for all monkeys and other non-human primates and humans also, then she finds something rather interesting. So these are rodents here, smoky-tailed shrew and mouse and hamster and so on. And you can see that they have relatively small number of neurons, 200 million or so. Guinea pig again, 240 million. Now when you start going to primates, you have 1,500 million from the owl monkey. The capybara, as I said, is got a very large brain by weight compared to the owl monkey, but similar neurons. The squirrel monkey has got 3,000 million neurons. The capuchin monkey also close to 3,000, close to 4,000. The macaque monkey has got close to 6,000 million neurons, and we have about 86 billion neurons for 1.5 kg. So we have a very, very large number of neurons. And this is again more than our other primate cousins, gorillas and chimpanzees and so on. So in other words, we have somehow got a large number of neurons, and one of the big mysteries is how did we get that large number of neurons? One view was that before about how, one view is that we have a brain much larger for our body size than other non-human primates like gorillas and chimpanzees and so on. That doesn't seem to be the case. Our brain size has scaled proportionately, but our neuronal number has increased rather substantially. So we can say that we are a primate with the largest number of nerve cells, neurons. But how that number, why that means that we have capacity for language, where does consciousness come from, how does one, have the capacity to create all these buildings and aeroplanes and so on, all of that is not yet clear. How the nerve cells put together have this kind of a dramatic function, and how cultural evolution has taken over because of the behavior function. One point I'd like to make is we started off by being survivors on this planet. We became the planet of the apes, but now we are the planet of the apps because we have generated such extraordinary tools that we can transcend properties of our body and function in extraordinary things. So, to end, the African elephant, for example, has got a much larger brain than we have. Does it have more neurons? We don't know, no one has checked that, but the guess is unlikely that it has more glial cells, but not as much neurons as we have, but that experiment needs to be done. Similarly with whales, we don't know that. But we are certainly unique amongst primates, whether in terms of neuronal number, whether that is the case for all animals, seems possible, likely, but that needs to be tested. So, to end, we've understood enormously about what the building blocks of neurons are. We understood how they're put together and how they connect, how small circuits function. We have from the outside in, top down, a great understanding of how big circuits function, but we know very little about how multiple layers of circuits put together interact with the outside world and how these bigger circuits function. And more importantly, we know very little about the software of the brain, how that's put together, language, abstraction, consciousness, and how this comes about. But there are, used to be a lot of theories about these kinds of things, but now the experimental tools also allow us to test which theories are valid and which are not, to a much greater level than ever before. So, the tiny bacterial cell, we know a lot, and there's much to learn there. And there's much to learn from our kind of life, from these kinds of lives. So all science must be encouraged so that you don't know where surprises come from and where the knowledge will come from. And if you want to understand this kind of a complexity as a brain, you also need to understand simple organisms, so-called simple organisms. Thank you very much.
ಥ್ಯಾಂಕ್ ಯು ಸರ್. ಈಗ ಬಹಳ ಕಷ್ಟದ ಕೆಲಸ ನನಗೆ ಕೊಟ್ಟಿದ್ದಾರೆ ಕನ್ನಡದಲ್ಲಿ ಸಮ್ಮರಿ ಮಾಡಬೇಕು. ಮೊದಲಿಗೆ ನಾವು ಪ್ರತಿಯೊಂದು ಸ್ಲೈಡ್ ಅನ್ನು ತೋರಿಸ್ತಾ ಅದ್ರ ಜೊತೆಗೆ ಕನ್ನಡದಲ್ಲೇ ಮಾಡಬೇಕು ಅಂತ ಅಂದ್ಕೊಂಡಿದ್ವಿ. ಆದ್ರೆ ಅದಕ್ಕಿಂತ 10 ನಿಮಿಷ ಸಮ್ಮರಿ ಕೊಡೋದು ಒಳ್ಳೆಯದು ಅಂತ ಅನ್ನಿಸ್ತು. ಹಾಗಾಗಿ ಹೀಗೆ ಮುಖ್ಯವಾಗಿ ಎಲ್ಲಿ ಅವನ ಗಲಾಟೆ ಮಾಡ್ತಿದ್ದ ಈ ಬೆರಳು ಈ ಬೆರಳು ಅದೆರಡರಲ್ಲಿ ಮಾತ್ರ ಅದನ್ನು ತೆಗಿಬೇಕು. ಈ ಬೆರಳು ಉಪಯೋಗಿಸಬಾರ್ದು. ಬರೀ ಈ ಬೆರಳು ಮಾತ್ರ. ಆದ್ರೆ ನಾವು ದೊಡ್ಡವರು ಮಾಡೋದಕ್ಕೆ ಕಷ್ಟ. ಮನುಷ್ಯರಿಗೆ ಕೆಲವು ವಿಶೇಷವಾದಂತ ಗುಣಗಳಿವೆ. ಮೊದಲನೇದಾಗಿ ಯೋಚನೆ ಮಾಡಬಹುದು, ನಮ್ಮ ಚಿಂತನೆ ಕಲ್ಪಿಸ್ಕೊಳ್ಬಹುದು, ಬೇಕಾದಷ್ಟು ವಿಷಯಗಳನ್ನ ನಾವು ಕಲ್ಪಿಸ್ಕೊಳ್ಬಹುದು. ಆ ಕಲ್ಪನೆಗಳಿಗೆ ನಾವು ಒಂದು ಮೂರ್ತ ರೂಪವನ್ನು ಕೊಟ್ಟು ಭಾಷೆಯಲ್ಲಿ ಇನ್ನೊಬ್ಬರಿಗೆ ಹೇಳಬಹುದು. ಇವೆಲ್ಲಕ್ಕೂ ಕೂಡ ನಮ್ಮ ಮಿದುಳು ಕಾರಣ ಅಂತ ಅನ್ನೋದಿದೆ. ಈ ಮಿದುಳು ಅಷ್ಟೊಂದು ಜಟಿಲವಾಗಿರುವಂತಹ ಒಂದು ಅಂಗವನ್ನ ನಾವು ಅರ್ಥ ಮಾಡ್ಕೊಳ್ಳೋದು ಹೇಗೆ ಅಂತ ಅನ್ನೋದು ಮುಖ್ಯವಾಗಿ ಮಿದುಳಿನಲ್ಲಿ ಯಾವ ರಚನೆಗಳಿವೆ ಅಂತ ನಾವು ನೋಡಿದ್ರೆ, ಮನುಷ್ಯನ ಮಿದುಳಿನಲ್ಲಿ ಕೋಟ್ಯಾಂತರ ನರಕೋಶಗಳಿವೆ. ಈ ನರಕೋಶಗಳಲ್ಲಿ ಮುಖ್ಯವಾಗಿ ಅದರ ಕೋಶ ಅಂತ ಅನ್ನೋದು ಒಂದು, ಮತ್ತು ಆ ಕೋಶ ಬೇರೆ ಕೋಶಗಳ ಜೊತೆಗೆ ಸಂಪರ್ಕ ಮಾಡಿಕೊಳ್ಳುವಂತಹ ನರತಂತು ಒಂದಿರುತ್ತೆ, ಅದನ್ನ ಆಕ್ಸಾನ್ ಅಂತ ಹೇಳ್ತೇವೆ. ಮತ್ತೆ ಅದೇ ರೀತಿಯಲ್ಲಿ ಬೇರೆ ನರಕೋಶಗಳಿಂದ ಬರುವಂತಹ ಆಕ್ಸಾನ್ಗಳ ಜೊತೆಗೆ, ನರತಂತುಗಳ ಜೊತೆಗೆ ಬೆರೆತುಕೊಳ್ಳೋದಕ್ಕೆ ಸಿನಾಪ್ಟಿಕ್ ಅಂತ ನಾವು ಕನೆಕ್ಷನ್ಸ್ ಅಂತ ಹೇಳ್ತೇವೆ, ಅವುಗಳ ಜೊತೆಗೆ ಇರ್ತವೆ. ಇದು ಒಂದು ನರಕೋಶ ನೂರಾರು ನರಕೋಶಗಳು, ಸಾವಿರಾರು ನರಕೋಶಗಳ ಜೊತೆಗೆ ಈ ರೀತಿ ಸಂಪರ್ಕ ಮಾಡ್ಕೊಂಡಿರುತ್ತೆ. ಒಂದರದನ್ನ ನಾವು ಅರ್ಥ ಮಾಡ್ಕೊಳ್ಳೋದು ಸುಲಭ, ಮತ್ತೆ ಇಷ್ಟೊಂದು ಮಲ್ಟಿಪಲ್ ಇದೆಯಲ್ಲ ಅವನ್ನ ನಾವು ಹೇಗೆ ಅರ್ಥ ಮಾಡ್ಕೊಳ್ಬಹುದು ಅಂತ ಅನ್ನೋದು ಒಂದು ದೊಡ್ಡ ಸಮಸ್ಯೆ, ಅದರಲ್ಲೂ ನಮ್ಮ ಜೀವವಿಜ್ಞಾನದಲ್ಲಿ. ಇದನ್ನ ಅರ್ಥ ಮಾಡ್ಕೊಳ್ಳೋದಕ್ಕೆ ಮೊದಲನೇದಾಗಿ ಒಂದು ಕಲ್ಪನೆ ಏನಂತಂದ್ರೆ, ನಮ್ಮ ಪ್ರತಿಯೊಂದು ಅಂಗಗಳ ಸಂಯೋಜನೆ, ಆರ್ಗನೈಸೇಷನ್ ಅಂತ ಏನ್ ಹೇಳ್ತೀವಿ, ಅವುಗಳು ಒಂದೇ ರೀತಿಯ ವಿನ್ಯಾಸ ಎಲ್ಲ ಜೀವಿಗಳಲ್ಲಿಯೂ ಇರಬೇಕು. ಅಂದ್ರೆ ನಾವು ಒಂದು ಕಟ್ಟಡ ಕಟ್ಟಬೇಕಾದ್ರೆ ಉಪಯೋಗಿಸುವಂತಹ ಎಲ್ಲ ಸಾಮಾನುಗಳು ಅಥವಾ ವಸ್ತುಗಳು ಕೂಡ ಒಂದೇ ರೀತಿ ಇರ್ತವೆ, ಆದ್ರೆ ಅವುಗಳ ವಿನ್ಯಾಸ ಮಾತ್ರ ಬೇರೆ ಆಗುತ್ತೆ. ಅವುಗಳಲ್ಲಿರುವಂತಹ ರಚನೆಗಳು ಬೇರೆ ಆಗ್ತವೆ, ಆರ್ಕಿಟೆಕ್ಚರ್ ಬೇರೆ ಆಗ್ತದೆ. ಅದೇ ಕಲ್ಲನ್ನು ಉಪಯೋಗಿಸಿ ನಾವು ಬೇಲೂರು ಹಳೆಬೀಡು ಕಟ್ಟಿರ್ತೀವಿ, ಅದೇ ಕಲ್ಲನ್ನು ಉಪಯೋಗಿಸಿ ನಾವು ಬೆಂಗಳೂರಿನಲ್ಲಿ ಪ್ಯಾಲೇಸ್ ಕೂಡ ಕಟ್ಟಿರ್ತೀವಿ. ಸೊ ಹಾಗಾಗಿ ಹೇಗೆ ಈ ರೀತಿ ವ್ಯತ್ಯಾಸ ಬಂತು ಅಂತ ಅನ್ನೋದನ್ನ ಮಾಡೋದಕ್ಕೆ ಮೂರು ವಿಧದ ಯೋಚನೆಗಳಿವೆ. ಒಂದು, ಬಹುಶಃ ಆ ಬೆಳವಣಿಗೆ ಹಂತದಲ್ಲಿ ಯಾವುದೋ ಒಂದು ವಿನ್ಯಾಸ ಒಳಗಡೆನೆ ಅಡಗಿರಬೇಕು, ಅದನ್ನ ಜೆನೆಟಿಕ್ ಅಂತ ಹೇಳ್ತೀವಿ, ಆಂತರಿಕವಾಗಿ ಇರಬೇಕು. ಅಥವಾ ಎರಡನೇದಾಗಿ ಅವು ಅಕ್ಕಪಕ್ಕದಲ್ಲಿರುವಂತಹ ಪರಿಸರದಲ್ಲಿ ಇರುವಂತಹ ವಸ್ತುಗಳಿಂದ ಪ್ರಭಾವಿತವಾಗಿರಬೇಕು. ಅಂದ್ರೆ ಇದನ್ನ ನಾವು ಒಂದು ಜೀವಕೋಶ ಬೆಳೀತಾ ಇರಬೇಕಾದ್ರೆ ಸುತ್ತಮುತ್ತ ಇರುವ ಜೀವಕೋಶಗಳು ಅದನ್ನ ಪ್ರಭಾವಿಸಿ ಏನಾಗಬಹುದು ಅಂತ ಅನ್ನೋದನ್ನ ತೀರ್ಮಾನಿಸ್ತವೆ ಅಂತ ಅನ್ನೋದು. ಮೂರನೇದು, ಅದು ಬೆಳೆಯುವಾಗ ತಾನೇ ಒಂದು ಬದಲಾವಣೆಗಳನ್ನ ಮೈಗೂಡಿಸ್ಕೊಳ್ಳುತ್ತೆ. ಈ ಅಧ್ಯಯನಗಳನ್ನ ನಾವು ಬೇರೆ ಬೇರೆ ರೀತಿಯಲ್ಲಿ ಮಾಡ್ತಾ ಇದ್ದೀವಿ. ಮುಖ್ಯವಾಗಿ ನಾವು ಮನುಷ್ಯನ ಮಿದುಳನ್ನ ಅಧ್ಯಯನ ಮಾಡೋದಕ್ಕೆ ಸಾಧ್ಯ ಆಗದೆ ಇರುವುದರಿಂದ, ಅಷ್ಟು ಸುಲಭ ಅಲ್ಲದೆ ಇರೋದ್ರಿಂದ, ಸರಳವಾದ ಜೀವಿಗಳಲ್ಲಿ ಏನಾಗ್ತಾ ಇದೆ ಅಂತ ನೋಡ್ಕೊಂಡು ಅದೇ ನಿಯಮಗಳು ಕೂಡ ನಮ್ಮ ಮನುಷ್ಯನ ಇದರಲ್ಲೂ ಆಗ್ತಾ ಇದೆಯಾ ಅಂತ ಅನ್ನೋದನ್ನ ನಾವು ಪರೀಕ್ಷೆ ಮಾಡ್ತೇವೆ. ಹಾಗೆ ಮಾಡೋದಿಕ್ಕೆ ಹೋದಾಗ ಮೊದಲನೇದಾಗಿ ನಾವು ಸ್ಕ್ವಿಡ್ ಅಂತ ಏನ್ ಹೇಳ್ತೀವಿ, ಆ ತರದ ನರಕೋಶವನ್ನು ನೋಡಿದ್ವಿ. ಯಾಕಂದ್ರೆ ಅದು ಅತ್ಯಂತ ಸರಳವಾದಂತಹ ಒಂದು ಮಿದುಳು, ಆದ್ರೆ ಬಹಳ ಉದ್ದವಾದಂತಹದ್ದು, ಸುಲಭವಾಗಿ ನಮಗೆ ಕಾಣುವಂತ. ಅದರಲ್ಲಿ ಕಂಡಾಗ ಒಂದು ನರಕೋಶದಿಂದ ಹೊರಟಂತಹ ವಿದ್ಯುತ್ತು ಆ ನರತಂತುವಿನ ಮೂಲಕ ಹೋಗಿ ಇನ್ನೊಂದು ನರಕೋಶವನ್ನ ಬಾಧಿಸುತ್ತೆ. ಗೊತ್ತಾಯ್ತು? ಅಂದ್ರೆ ವಿದ್ಯುತ್ ಯಾವ ರೀತಿಯಲ್ಲಿ ಹುಟ್ಟುತ್ತೆ, ಅದಕ್ಕೆ ಕಾರಣ ಏನು, ಮೂಲ ಕಾರಣ ಏನು ಅಂತಂದ್ರೆ ಹೊರಗಡೆ ಇರುವಂತಹ ಲವಣಗಳು ಒಳಗಡೆ ಹೋಗುವಂತದ್ದು, ಹೊರಗಡೆ ಬರುವಂತದ್ದು, ಈ ರೀತಿಯಲ್ಲಿ ಗೊತ್ತಾಯ್ತು. ಇವೆಲ್ಲವನ್ನ ಅಧ್ಯಯನ ಮಾಡಿಕೊಂಡು ಮುಂದೆ ಹೋದಾಗ ಬೆಳವಣಿಗೆಯ ಹಂತದಲ್ಲಿ ಪ್ರತಿಯೊಂದು ಜೀವಿಯಲ್ಲಿಯೂ ಕೂಡ ಈ ನರಕೋಶಗಳು ಹೇಗೆ ಬೇರೆ ಆಗ್ತವೆ, ಒಂದೇ ವಸ್ತು ಆದ್ರೂ ಕೂಡ ಹೇಗೆ ಬೇರೆ ಆಗ್ತದೆ ಅಂತ ಕಾಣೋದಕ್ಕೆ ಹೋದಾಗ ಸ್ಟೆಮ್ ಸೆಲ್ ನ ಕಾನ್ಸೆಪ್ಟ್ ಅನ್ನ ಕೂಡ ನಾವು ಬಳಸ್ತೀವಿ. ಅಂದ್ರೆ ಸ್ಟೆಮ್ ಸೆಲ್ ಅಂತ ಅನ್ನೋದು ಇರ್ತದೆ, ಇದನ್ನ ಆಕಾರ ಕೋಶ ಅಂತ ಹೇಳ್ತೇವೆ, ಕಾಂಡಕೋಶ ಅಂತ ಹೇಳ್ತೇವೆ, ಸ್ಟೆಮ್ ಸೆಲ್ಸ್ ಅಂತ ಕೂಡ ಹೇಳ್ತೇವೆ. ಇದು ಒಂದು ಒಂದಾಗಿ ಅದು ಇನ್ನೊಂದನ್ನು ಹುಟ್ಟಿಸ್ಕೊಂಡಾಗ ಎರಡನೆಯದರಲ್ಲಿ ಒಂದು ಸ್ಟೆಮ್ ಸೆಲ್ ಆಗಿ ಉಳ್ಕೊಳ್ತದೆ, ಮತ್ತೊಂದು ಇನ್ನೊಂದು ಸ್ವಲ್ಪ ವ್ಯತ್ಯಾಸ ಆಗ್ತದೆ. ಹೀಗೆ ಮತ್ತೆ ತಿರುಗಿ ಹುಟ್ಟಿದಾಗ ವ್ಯತ್ಯಾಸ ಆಗುತ್ತೆ. ಆ ವ್ಯತ್ಯಾಸಗಳು ಯಾವ ರೀತಿಯಲ್ಲಿ ಜೋಡಿಸ್ಕೊಂಡು ಹೋಗ್ತವೆ ಅನ್ನೋದ್ರ ಮೇಲೆ ಬಹಳಷ್ಟು ವ್ಯತ್ಯಾಸಗಳನ್ನ ಆಗ್ತವೆ ಅಂತ ಅನ್ನೋದು ಒಂದು ಕಾನ್ಸೆಪ್ಟ್. ಇದನ್ನ ಪರೀಕ್ಷೆ ಮಾಡೋದಕ್ಕೋಸ್ಕರವಾಗಿ ಹಲವಾರು ಪ್ರಯೋಗಗಳನ್ನ ಮತ್ತೊಂದನ್ನ ಮಾಡಿರ್ತಾರೆ. ಅವುಗಳ ಬಗ್ಗೆ ವಿವರಣೆಯನ್ನ ಪ್ರೊಫೆಸರ್ ವಿಜಯರಾಘವನ್ ಕೊಟ್ಟರು. ಒಟ್ಟಾರೆ ಒಂದು ಕಾಲದಲ್ಲಿ ನಾವೇನ್ ಅನ್ಕೊಂಡಿದ್ದೀವಿ, ಮನುಷ್ಯನ ಮಿದುಳು ಬಹಳ ದೊಡ್ಡದಾಗಿರೋದ್ರಿಂದ, ನಮ್ಮ ದೇಹದ ಗಾತ್ರಕ್ಕೆ ಹೋಲಿಸಿದಾಗ ಅದು ಬಹಳ ದೊಡ್ಡದಾಗಿರೋದ್ರಿಂದ ಅದು ಭಿನ್ನವಾಗಿದೆ ಅನ್ಕೊಂಡಿದ್ವಿ. ಆದ್ರೆ ಇವತ್ತಿನ ಲೆಕ್ಕದಲ್ಲಿ ಈ ರೀತಿಯ ಅಧ್ಯಯನಗಳು ಪ್ರತಿಯೊಂದು ಜೀವಿಗಳಲ್ಲೂ ನಾವು ಮಾಡೋದಕ್ಕೆ ಹೋದಾಗ ಬರೀ ಅದೊಂದೇ ಕಾರಣ ಇರಲಿಕ್ಕಿಲ್ಲ. ಅದರ ಜೊತೆಗೆ ಮಿದುಳಿನಲ್ಲಿ ಇರುವಂತಹ, ನಾವು ಗ್ಲಿಯಲ್ ಸೆಲ್ಸ್ ಅಂತ ಏನ್ ಹೇಳ್ತೀವಿ, ಅವು ಮತ್ತು ನರಕೋಶಗಳು, ಇವೆರಡರ ಮಧ್ಯದಲ್ಲಿ ಇರುವಂತಹ ಪ್ರಮಾಣ ಅದು ಕೂಡ ಬಹಳ ಮುಖ್ಯ ಆಗ್ತದೆ. ಮನುಷ್ಯರಲ್ಲಿ ಈ ನರಕೋಶಗಳ ಸಂಖ್ಯೆ ಆ ಗಾತ್ರಕ್ಕೆ ಹೋಲಿಸಿದ್ರೆ ಬಹಳ ಹೆಚ್ಚು ಅಂತನ್ನೋದು ಈಗ ನಮಗೆ ಗೊತ್ತಿರುವಂತದ್ದು. ಇಲ್ಲಿಯವರೆಗೆ ನಮಗೆ ಆ ನರಕೋಶಗಳನ್ನು ಒಂದೊಂದನ್ನಾಗಿ ಹುಡುಕಿ ಅಧ್ಯಯನ ಮಾಡೋದಕ್ಕೆ ಆಗ್ತಿರ್ಲಿಲ್ಲ. ಆದ್ರೆ ಈಗ ಇರುವಂತಹ ಸಾಧನಗಳು, ತಂತ್ರಗಳು ಅವನ್ನ ಮಾಡೋದಕ್ಕೆ ಸಾಧಿಸಿಕೊಟ್ಟಿವೆ. ಹಾಗಾಗಿ ನಾವು ಈಗ ಇಷ್ಟರ ಮಟ್ಟಿಗೆ ತಿಳ್ಕೊಂಡಿದ್ದೇವೆ. ಅಂದ್ರೆ ಮನುಷ್ಯನ ಮಿದುಳಿನಲ್ಲಿ ಆಲೋಚನೆಗಳು ಬರೋದಕ್ಕೆ, ನಾವು ಕನಸು ಕಾಣೋದಕ್ಕೆ, ಕಲ್ಪಿಸ್ಕೊಳ್ಳೋದಕ್ಕೆ, ಹೊಸ ವಿಚಾರಗಳನ್ನ ತಿಳಿಯೋದಕ್ಕೆ, ಮಾತಾಡೋದಕ್ಕೆ, ಇನ್ನೊಬ್ಬರಿಗೆ ತಿಳಿಸ್ಕೊಡೋದಕ್ಕೆ, ಇದಲ್ಲದೆ ಹೊಸ ಹೊಸ ವಿಷಯಗಳನ್ನ ಕಟ್ಟೋದಕ್ಕೆ, ಇವೆಲ್ಲಕ್ಕೂ ಕೂಡ ಮುಖ್ಯವಾಗಿ ಈ ಅತಿ ಹೆಚ್ಚಿನ ನರಕೋಶಗಳು ಇರೋದು ಕಾರಣ. ಆದರೆ ಅದು ಹೇಗೆ ಈ ಎಲ್ಲಕ್ಕೂ ಕಾರಣವಾಗಿವೆ ಅಂತ ಅನ್ನೋದು ನಮಗೆ ಇನ್ನೂ ಗೊತ್ತಿಲ್ಲ. ಅದು ಮುಂದಿನ ಹೆಜ್ಜೆ ಆಗ್ಬೇಕು. ಇಷ್ಟುವರೆಗೆ ನಾವು ದಶಕಗಳು ಮತ್ತೆ ಶತಮಾನಗಳಿಂದ ನಾವು ಕಲ್ತ್ಕೊಂಡು ಬಂದಿರೋದು ಇಷ್ಟು. ಇವತ್ತಿಗೆ ನಮಗೆ ಗೊತ್ತಿರುವಂತದ್ದು, ಬೇರೆ ಎಲ್ಲ ಪ್ರಾಣಿಗಳಿಗಿಂತ ನಾವು ಯಾಕೆ ಭಿನ್ನ ಅಂದ್ರೆ ನಮ್ಮಲ್ಲಿ ನರಕೋಶಗಳ ಸಂಖ್ಯೆ ಜಾಸ್ತಿ ಇದೆ. ಆದರೆ ಅವುಗಳ ಸಂಯೋಜನೆಯಲ್ಲಿ ಯಾವ ರೀತಿಯಲ್ಲಿ ನಮ್ಮಲ್ಲಿ ಇರುವಂತಹ ಸಾಮರ್ಥ್ಯಗಳು ಹುಟ್ಟುಕೊಳ್ತವೆ ಅನ್ನೋದು ನಮಗೆ ಇನ್ನೂ ಸ್ಪಷ್ಟವಾಗಿ ಗೊತ್ತಿಲ್ಲ ಅಂತ. ಇವಿಷ್ಟು ಅವರು ಹೇಳಿದ್ದು. ಈಗ ಪ್ರಶ್ನೆಗಳು.
ಕನ್ನಡದಲ್ಲಿ ಕೇಳಬಹುದು, ಇಂಗ್ಲಿಷ್ ಅಲ್ಲಿ ಕೇಳಬಹುದು, ಕನ್ನಡದಲ್ಲಿ
just to shut up. It was a good presentation. So just couple of questions are the doubts what I had. So right from my childhood or anyone, there is a myth that brain, human brain is used only up to maximum 10%. Whether that is true or, because a lot of articles have that, someone says it is true, someone says it is not true. So what is the real fact? So that is one question. Second one is, as we grow, as we are human is born, as we mature enough, whether brain develops and attains a maturity, and as we get older, again it diminishes. So what is the growth of a brain as such?
Yeah, about the first one, it is indeed a myth, because that came from a view, because most of the human brain are non-neuronal cells, glial cells, and people thought they are not used normally, so the brain is not used. It's not correct to say that the glial cells are not used, but they're not used from our traditional understanding of brain function. The rest of the nerve cells are used, so it's not 10%. It will be, I would say, there are ways by which, when parts of the brain are damaged, other parts compensate. But it's not that those parts are not normally used. So almost all the brain is used, but there's an ability to compensate even upon damage. Second part, of course, as one ages, goes beyond puberty, and beyond, adolescence and beyond, the numbers of nerve cells start declining. They start dying in large numbers. So as you grow older, the numbers of nerve cells die. As I've told you, there are a very large number of nerve cells, and that kind of damage doesn't necessarily correlate with cognitive decline. There are no shortage of examples of loss of nerve cells without cognitive decline, and that's not necessarily causative. Of course, it does affect many other aspects of functioning, but yes, nerve cells do decline with age.
So what is the memory capacity of the human brain cell with respect to petabytes? From where I get petabytes are there, the memory capacity of the human brain. Usually hippocampus is there for short-term memory and long-term memory. And finally, what is the memory capacity of the human brain?
So, unfortunately, I can't give you the numbers. I mean, they are available.
Approximately.
you can check that. But importantly, there are two factors. One, to have the kind of storage capacity of one human brain in terms of information uh, and analysis, you need very huge computers which have got extraordinarily high power requirements, whereas the biological power requirements are much, much smaller. Secondly, and more important than memory amount itself, the amount of memory which you can store, are speed of recall and reuse across different parts of the brain, different modalities. That's incredibly fast. So we don't know yet whether a digital system can act so rapidly and store such information across a whole range. It can do a really incredible job in certain narrow areas, just as a fruit fly can fly and we cannot fly. There are computers which can store and act relatively rapidly in very specific areas, playing chess or doing a specific task and so on. But this kind of a cross-spectrum capability which human brains have, and ability for intuition, abstraction, and so on, of analog circuits are yet not understood in the context of energy requirements, space requirements, and reform.
ಯಾಕೆ ಈಗ ವಿಶ್ವೇಶ್ವರಯ್ಯ ಅಥವಾ ಚಾಣಕ್ಯ ಅಥವಾ ಶಕುಂತಲಾ ದೇವಿ ಇವರೆಲ್ಲ ಮ್ಯಾಕ್ಸಿಮಮ್ ಆಗಿ ಬ್ರೈನ್ ನ ಬಳಸ್ಕೊಂಡು ಸಾಮಾನ್ಯ ಜನಕ್ಕೆ ಯಾಕಂದ್ರೆ ಸಾಧ್ಯ ಇಲ್ಲ ಅಥವಾ ನಾವು ಹೇಗೆ ಮ್ಯಾಕ್ಸಿಮಮ್ ಆಗಿ ಬ್ರೈನ್ ನ ಬಳಸ್ಬೋದು?
So that, let us take the example of people who can calculate very rapidly, like Shakuntala Devi. Right? Other examples are not so striking because they are very unusual people of a kind there are many other unusual people like that all over the world. But the ability to calculate numbers or have musical capabilities at a very young age is very, very unusual. And we don't know how that capability comes in terms of the brain. But we do know that, there are a very small number of people who have such extraordinary capabilities. John von Neumann, for example, was another person who was supposed to be a genius in a broader spectrum, which is very, very rare. One thought is that as we grow, typically what happens is that there are inhibitory neurons which keep us coordinated in a world. When you are a child growing up, you know every street shop name, every sign, every song which came, everything you notice. But as you grow older, when you go to the same street, you don't notice anything, right? Because a lot of survival in the world is about filtering out information and not keeping it. When the filtering capability is not fully there, then you have the ability to do things which are unusual, but it can also potentially, not in all cases, some cases, so we don't know that.
Sir,
Let's keep that one question after that, please.
Sir, we see on the books that the brain has half of divided cell, but here it is fully covered.
It is only one part.
So that's a good point. The brain has got two halves. So you asked a very important question. Each half of the brain is like a fully functional brain, but they are connected with each other. There's what is called a commissure which connects them, and the information transfer from one half to another is so fast that it behaves like one unit. If you could have that kind of an information transfer from me, from my brain, to speaking to you at such a fast rate, there's no gap at all, and that feeds back over here, then that is what happens in computers and what are called massively parallel computers. But we don't have that kind of a speed for interhuman communication like we have with intra-brain communication.
ನಮ್ಮ ಮಿದುಳೊಳಗೆ ಎರಡು ಪಾರ್ಟ್ ಇರುತ್ತಲ್ಲ, ಅದರ ಮಧ್ಯೆ ಒಂದು ಕನೆಕ್ಷನ್ ಇದೆ. ಆ ಕನೆಕ್ಷನ್ ನಲ್ಲಿ ಇದು ಬೇರೆ ಬ್ರೈನ್, ಇದು ಬೇರೆ ಬ್ರೈನ್ ಆದ್ರೂ ಕೂಡ ಅವೆರಡು ಕೂಡ ಒಂದು ಜೊತೆ ಒಂದು ತುಂಬಾ ಫಾಸ್ಟ್ ಆಗಿ ಮಾತಾಡ್ಬೋದು. ಹಾಗಾಗಿ ಎರಡೂ ಕೆಲಸ ಮಾಡುವಾಗ ಒಂದೇ ಬ್ರೈನ್ ರೀತಿಯಲ್ಲಿ ಮಾಡ್ತಾರೆ. ಅದು ಒಳಗೆ. ನನ್ನ ಮತ್ತೆ ನಿನ್ನ ಬ್ರೈನ್ ಮಧ್ಯ ಅದೇ ರೀತಿಯಲ್ಲಿ ಮಾಡೋದಕ್ಕೆ ಸಾಧ್ಯ ಆದ್ರೆ ತುಂಬಾ ಚೆನ್ನಾಗಿರ್ತಿತ್ತು. ನಾನು ಸುಮ್ಮನೆ ಇಷ್ಟು ಹೇಳಿದ್ರೆ ಪೂರ್ತಿ ನೀನು ಅರ್ಥ ಮಾಡ್ಕೊಂಡು ಹೋಗಿದ್ದು, ಪಾಠ ಮಾಡೋ ಅವಶ್ಯಕತೆ ಇಲ್ಲ. ಆದ್ರೆ ಕಂಪ್ಯೂಟರ್ಗಳಲ್ಲಿ ಪ್ಯಾರಲಲ್ ಕಂಪ್ಯೂಟರ್ಗಳಲ್ಲಿ ಅದು ಸಾಧ್ಯ ಆಗುತ್ತೆ, ನಮ್ಮಲ್ಲಿ ಇನ್ನೂ ಅದು ಸಾಧ್ಯ ಇಲ್ಲ. ಆದ್ರೆ ನಮ್ಮ ಎರಡು ಬ್ರೈನ್ಗಳು ಕೂಡ ಆ ರೀತಿ ಮಾತಾಡ್ತಾರೆ. ಇನ್ನೊಂದು
Sir, my sir told that when we use right hand to write or read for anything, our left brain will work, and when we use left hand, the right brain will work. Why it will not work right or the left?
So that happens, a very good point. That is partly because of the way the connection from the eyes and the body go to different parts of the brain. There are primary and secondary connections. In some organisms, such as human, the connection goes from, some of the major connections go from one side to another, and this side to the other, and then they also talk to each other. So there is a dominance of one side over the other because of that, and for other reasons. But it is not that there's completely one side is completely dominant or one side is completely specialized. Some people say music, arts are specialized in one side and science and engineering on another side of the brain. That's not correct. But the capabilities are there.
Sir, regarding the from first floor, sir. Sir, regarding the nerve cells or the neurons which do different functions, like there are two part of this question actually. One is when there is physical damage to our brain due to maybe some accident or injury or something like that. Is it possible that the neurons are trained by the rest of the brain, which I mean, the damaged part takes over, or second part is like whether the same portion of the brain, whatever work it does, one particular portion of the brain, if it is not able to function, can the other portion of the brain take over, or can it be trained by some method?
Yeah, but this is a very interesting and important question and it's been studied a lot. There are many kinds of responses to damage. One, which is very common, is if a very major part involved in a particular function is damaged, then you don't have that function. Right? If the optic lobe is very badly damaged, then your visual system is not working properly. The other is that when there is damage, there is some kind of compensation from neighboring regions. So this can happen sometimes rather dramatically in very severe damage also, right? For example, in strokes, or when there's a bullet or a rod which goes through, or because of people are born with large areas of the brain missing. Then there was one case recently of a woman who went to a hospital for something and they did an MRI and they found a very large part of the brain was absent, but she has grown up fully without noticing anything at all. So how did that happen? Is that because the damage was there right from development that the compensatory mechanisms could set in? There are other people who have got damages later on in life. There was a person who had there was a railway track engineer who had a big part of the track going through his brain and lost many capabilities, lost immediate short-term memory, but had long-term memory really well. So he thought that he was trapped, as he grew older, his every response was trapped, to the period before he had the accident. So there are those kinds of situations where the damage doesn't have to be extensive. And there are yet others, because of slow regeneration and training, there is some amount of compensation, and that's quite remarkable that that's being addressed. Finally, there are areas where the compensation takes place in very strange manners, and that's been addressed very beautifully both by Oliver Sacks in his many books, as well as V.S. Ramachandran in his books about phantom limbs and phantom phenomena. So, if you, for example, have motor nerves, sensory nerves going to a particular part of the brain, so you know that you're holding this, and if this is damaged, then the other parts of these nerve cells and the circuits go to some other parts of your body. So when you touch the chest, the brain seems to think that you're touching the arm, the missing arm. So, those kinds of strange things happen, phantom phenomena happen. Or you get pain in a limb which is not there. The limb is not there, so it shouldn't pain. But you say, oh, my wrist is paining, but there's no wrist for it to pain. So those are the kinds of very interesting things.
So recently I read some papers about vitamin D receptor and melatonin. Melatonin binds to vitamin D receptor and controls the function of vitamin D. Like circadian rhythm with the daytime and vitamin D function, and melatonin is directly associated with the nighttime.
Yeah, I'm not very sure about the effectiveness of melatonin and circadian rhythm. There are many views on that. I don't know what is the current state of science on that. But circadian rhythm have been very well studied. It's one of the few examples where the genetic basis of that impinges on behavior in a very direct manner. That's a long story, but there are genes which are more active in the daytime and go down, and there are other genes at night and go up, but they control the rhythm. And that relates in many other ways to other bodily functions, eating, sleeping, and so on. So that's a very important and exciting area.
And also like cancer biology stuff night shift workers, especially women, they get breast cancer more due to melatonin disruption.
Right. So that kind of a thesis that changes in circadian rhythm affect other functions and can cause, for example, lifestyle diseases is a very important area. There's a person at the Salk Institute, Sapsidanand Panda, who's doing some really nice work.
Right?
What do you think of difference in the brain you will find between ants and flies? Because you see behavior home, community living and all that. What is it that?
Well, that's a very interesting question. Ants fall into a good example because there are ants where whom the smell dominates. You can see that they have big antennae. There are other ants which have the visual system, the eye dominates. Some other ants have very small eyes. So, depending on the kind of ant, if you open up the brain, here you see this part of the brain in the fly is the optic lobe, right? Here, which is not so well shown, the lights are too bright, are the regions, here is a region where the smell is processed, over here, right? Now, in an ant which has very little visual activity in its function, this will be very, very small. And that will, those optic lobes, the antennal lobes will be much larger. So ants will change a lot. Flies use visual system enormously, right? And smell also. So their olfactory glomerulus and their antennal, their optic lobes are very, very large. With ants, it will vary a lot.
Behavior also changes because of.
Well, so evolutionarily, there is a interaction between having a behavior which allows survival in a particular environment and how the brain therefore has changed to allow that behavior to happen efficiently. How does this happen? We know a lot about that. I told you about stem cells making the brain. So there will be a very small number of divisions in an ant which has a very small optic lobe, right, of those stem cells, and a large number of divisions in an ant which has a large optic.
So the mathematical models, actually mathematicians, mathematical models are also constructed in respect of cognitive science.
Okay. With respect to?
In respect of cognitive science, mathematical models are also constructed with respect to study of human brain also. Whether these mathematical models are based on the experimental input or simply it is a mathematical model.
So, I mean, not just mathematical models, there are multiple theories of how, neural circuits function at higher level. That has led to computer science in those kinds of areas developing a lot, but not necessarily an increase in our understanding of how the brain itself works. In some cases, they are fed back to very valuable models of how the brain works, and those have been substantially in terms of how how information can. Yeah, I'm sorry, it looks like I've destroyed your microphone.
You can use that.
Yeah. So, mathematical models have been very effective in how nerve cells communicate with each other. That's been very valuable. In information content and information theory, they've been very valuable. They have not yet been so useful in revealing how mechanisms of higher order functions take place. There you're right. Testing them has been not easy till recently, but now better and better tests on this.
Sir, if I may, what are the top 3 problems which the brain scientists are now pursuing? Or is the question itself is?
No, this is a very important question. See, there are multiple parts of the brain which are coming together in helping our understanding. One is we know more and more about the nerve cell. We know how it communicates with other small nerve cells. We've got extraordinary tools to look at the physiology of function. What is earlier you had to poke electrodes inside. Now you can, without poking electrodes, by using fluorescent dyes, you can look at function of a large number of neurons. And it's not just like MRI saying this area is bright, that area is bright. In model organisms such as flies and mouse, you can say, I want to look at how these 50,000 neurons function and see whether what is happening. So the reagents to look at the anatomy, the physiology have become very good. And outside in the organism, the reagents to look at behavior of a freely moving organism, why you get this kind of information in many cases has become feasible. So data is no longer limited in our requirement for understanding. There are two major limitations, and I'll come to what the major questions are. One is the ability to even experimentally use all this data and behavior to predict behavior in a robust sense. Right? That is a big challenge. That challenge will have to come by a combination of experiment and theory. And theory is lagging far behind. We don't have good theories to be able to understand this big data in the context of the brain. We can do algorithms which will say, with this data, this is a predicted next step, but that is a more of a statistical analysis rather than an understanding. In that context, what are the big questions we should know? And that really is how are, first of all, the development of the brain, how does this development happen in a manner which is so precise? Why is it that despite the variability in connections, when a fruit fly is born, it can take off and fly as soon as it just comes out of the pupal case, it doesn't have to go to flight school. How is the capability to function in the outside world, either during development or soon after development, in our case in a year, how is that put together? That's a developmental question is one. The second question is an evolutionary one. How amongst all other organisms are our brains different or similar? How does that come together? How do our brains function in the context of other organisms? That question also is a very exciting question. Is there sentience? Is there cognition in other animals? And if so, how much? Again, the tools to measure that are very excellent. So, development, evolution, and the third is function. Function, I told you about how we function in a complex situation. Understanding that at a higher level. And function includes our ability for language, for communication, for consciousness. What are the material basis of that is something which, again, so I would say these are the three kinds of questions. But these three questions are what biologists ask again and again about everything. how is it made, where did it come from, how does it work? And they will always be asked, as you get more knowledge, you will keep on asking.
Sir, yeah. Is there a gender division in the brain?
So, this is a question which typically shouldn't be answered yes or no. Because it can be posed in a very different way. There are lots of areas in terms of development and sexual differentiation which are different amongst male and female in all organisms, and they function. That is because of the brain development and function being one component which is different. Hormonal differences are there, brain differences are there, and so on and so on. Now, the more fundamental question which many people ask is, do all these changes result in any differences in other aspects of function unrelated to the differences? And that the answer is no. I mean, so I can give you examples, but that answer is yes.
So my question is that as human beings, many of them when they get old, they will get Alzheimer's disease where they forget the things. So is there any more research? Like, is there any simulators which have been created in such a way, like if we lose our hand or leg, we have a artificial one. So like Alzheimer's where the connectivity won't be there. Is there anything which is made out of our head which has been connected and made it function?
Correct.
No, so I would say even before getting Alzheimer's or various kinds of age-related decline in cognitive functions, we have changed the world dramatically as humans. If you look at average IQ today compared to average IQ 100 years ago, and you normalize for social and economic and cultural context, IQ levels have gone up substantially. And the reason why that has gone up are for multiple reasons. The printing press, books, phones. You can ask me a question about, what is the memory capacity of the human brain and how fast it is. And I can say I don't have the numbers, but both you and I can look at the computer and get that information. 100 years ago, my wrong statement would have been taken as, someone from the podium giving a statement. That's not the case. So these tools allow us to function, these prosthetics allow us to function in our mental capacity much the same way as my spectacles or hearing aid or my walking stick allow me to function. And they are functioning at a million-fold greater efficiency than the walking stick. This allows me to see what is happening around the world, without having the richness or the wealth to do that. So we already do. Now, when actual physical decline of brain function takes place, are there treatments for that? There are. Very simply, slowing decline can be done by diet, exercise, activity of various kinds, and good luck. So it's not easy to have all of these. Sometimes you are unlucky, sometimes you don't eat properly, sometimes you don't do both. So that's the situation. Prosthetics for your knee replacement, hip replacement are much more straightforward. Brain, there are lots of efforts in regenerative biology and medicine. They have not made that much progress, though spinal cord regeneration is a major area where I think we'll start seeing modest to moderate successes. So let's see.
That will be the last question. After that, one question from my side.
Can we ask here?
So then two questions. Okay.
Someone from upstairs also.
Training ಅಂತ ಹೇಳಿ ಮಾಡ್ತಾರಲ್ಲ,
Brain.
ಟ್ರೈನಿಂಗ್ ಇದು ರಿಸರ್ಚ್ ವಯಸ್ಸುಗಳು ಅವ್ರು ಮಾಡ್ತಾರೆ. ಯಾಕೆಂದ್ರೆ ಚಿಕ್ಕ ಮಕ್ಕಳಲ್ಲಿ ಅದರ ಬ್ರೈನ್ ಡ್ಯಾಮೇಜ್ ಆಗೋ ಸಾಧ್ಯತೆ ಇದೆ. ಬಟ್ ಅದನ್ನ ಕನ್ಸಿಸ್ಟೆನ್ಸಿ ಮೇಂಟೈನ್ ಮಾಡ್ತಾರೆ.
Training is important, but there are also some myths about that. So, doing Sudoku will not help you becoming a better bank manager. So, practicing what bank manager has to do will make you a better bank manager. Practicing Sudoku will make you better at Sudoku. Practicing chess will make you better at chess. So unfortunately, because of the specialization of different parts, I mean, there'll be some general value, but it is not such a high level of general value as sometimes people think. If I lift weights with one arm, I will not become a better sprinter or a long distance runner. To strengthen my arm, I have to lift weights for the arm. For long distance, I have to do long distance training. For sprinting, I have to do that. It's work. And the same is true for different parts of the brain. Now, developmentally, there are some barriers to learning. That is, you can learn music, art. I joke that I can follow Kannada, my wife can speak Kannada, my son can read Kannada. But if I were growing up here, I might have been able to do all of these three without any problem, right? Now I find it very difficult to read Kannada. I have to keep on trying. I bought this, learn Kannada in 30 days. I bought it 30 years ago. But I still haven't know how to write. But a kid can learn it in no time. So there are some developmental times where you can learn much faster. Is it unsurmountable? No. If I'm in deep trouble, I've learned Kannada in 30 days. Right? That's not the problem. But it is a difficult task. So there are age-related barriers. Therefore, composite training early on is very important.
Some people will say that learning Veda in the chanting mantras helps to memorize everything. Memory power will increase. Is it true?
That's an interesting point there. It's not just the Vedas.
So,
It's not the Vedas.
our teachers will, one period they will teach.
No. So there was a the study of people who memorize large volumes of data and shown whether it is a London taxi driver or those who memorize and recite the Vedas or other kinds of huge volumes, their hippocampal size in one region, I don't remember which part of the brain specifically, was shown to be larger. Now, that was also been shown in flies if you are really adept at certain kinds of habituation. So that's because of increased sprouting of connections. Now, that is a consequence of activity of one part of the brain, and that's increased connectivity in that part of the brain. Whether that is more valuable for other parts, I don't know. So nowadays everyone has a Google map, but the London taxi drivers were famous. If you tell them a particular street, they had this instant recall and they will know where it is and they will go, right? And I'm sure our auto rickshaw drivers also had that kind of capability. Now they say location could, and both of you and them are looking at the map and going. So I'm not sure whether that has any practical impact in other areas.
Sir, nowadays youngsters do a lot of multitasking. Is our brain capable of doing it, or we are putting more pressure on it?
I mean, certain extent of multitasking is feasible. It's also of survival value. You cannot focus so much on something that you ignore. People do, sometimes they're too focused. If you're focused on your phone while crossing the street or on the motorcycle like this, that multitasking is very dangerous. But overall, focusing on a task is better than doing too many things at the same time.
So, one question here.
Last question.
Would you say there are some, or are there design flaws or disadvantages to the way our brain functions, develops, or is structured?
Disadvantages?
Yeah. Some cons.
The challenge in evolution is like a joint family house. You build something, then you modify it and you build something on top of that. It is not like demolishing a house and building a new apartment complex. So as a consequence of that, and another important feature, evolution is just so. It is not, survival of the fittest. It is survival. If you can manage to survive, you're okay. So you find a lot of redundancies, bad design, everything. But over a long period of hundreds of millions of years, you find extraordinary things happen. If you were to start again, would you design the same way? Would nature design it the same way? We don't know. But importantly, from the perspective of the brain, this shoddy design, this imperfect computation has resulted in the ability for change and abstraction as opposed to focusing only on precision and that capability, right? That has given an enormous flexibility and a computing capacity far in excess of what the original selections were. The original, capabilities for which our brain was evolutionarily selected did not involve the ability to do algebraic geometry or to make forms, but it can do that because of other kinds of redundancies which are there and the software which is on top of it. So yeah, it is badly designed, but it functions enormously well.
Yeah, that's the last question. Thank you. Thank you. So my question is more related to healing. So how much is there enough scientific proof that that our intentional positive thinking has a positive impact on healing any kind of ailments in the body? Does our brain have that capacity that it really changes based on our intentional thinking?
Okay, so about a couple of decades ago, such a question would have been dismissed as being very speculative. Over the last few decades, there have been more and more information about how neurohormonal and neuroimmunological interactions work, and also the other way around, how immunological and vascular inputs affect the brain function. So, not unexpectedly from a biological point of view, these systems interact a lot. Are they sufficient or not? Are they significant or not? Depends on the context. In some contexts, an extraordinary stress might tilt the balance of healing in a manner where it's not easy to happen. In another context, the extent of damage may be so severe that it doesn't matter whether you're stressed or not. It might just improve your mental state and your happiness, so it's okay. Even if you have a very serious illness, to not worry about it too much. Not because not worrying helps you heal, but worrying doesn't help. So it's a spectrum. But there are many situations now where clearly stress can be negative in the healing process.
So last question from my side. I heard that you had your BTech degree from IIT Khan, and this is biology. So there is a connection. Graduate degree from IIT Kanpur, and what you have done, achieved, is in biology. How is the connection?
Okay, I mean, there are 2 points. One is it's true that biology in school or in college. It is only because of ignorance that I jumped into something. If I had known biology or in college, it is only because of ignorance that I jumped into something. If I had known biology, I would have been very scared to do biology because it is so complex. Engineering is much more simple. So I jumped into that. Now, doing well in a subject is not so much your intrinsic capability. Is not so much your intrinsic capability. Of course, your motivation should be there, but it's really about the institutional environment and the people, right? The Tata Institute of Fundamental Research, where I studied, was an extraordinary place, and that place, Homi Baba said, it is easy to have a place where great people do great work, but a great institution is one where ordinary people can do great work, right? And that is what the Tata Institute is, and many of the government institutions which have been started are top research institutions, allow ordinary people to do great work. Also, there are extraordinary people coming in, really top quality students, top quality postdoctoral fellows, colleagues from here and all over the world. So just as I told you the importance of, you know, your phone or your walking stick or your bicycle or your car as prosthetics which help you, your community of friends, there is nothing more important than that. for intellectual pursuits. There is very little you can do on your own, but there is nothing which you cannot do by interacting with others.
Thank you, Professor Vijayaraghavan. It's now time for us to say thanks to you. There are two things. ಕನ್ನಡದಲ್ಲಿ ಹೇಳ್ಬಿಡ್ತೇನೆ. ಪ್ರೊಫೆಸರ್ ವಿಜಯರಾಘವನ್ ಅವರನ್ನು ನಾನು 3 ವರ್ಷದ ಹಿಂದೆ CFTR ನಲ್ಲಿ ಇದ್ದಾಗ ಭೇಟಿಯಾದೆ. ಆಗ ಅವರು ಪ್ರಿನ್ಸಿಪಲ್ ಸೈಂಟಿಫಿಕ್ ಅಡ್ವೈಸರ್ ಟು ಗವರ್ಮೆಂಟ್ ಆಫ್ ಇಂಡಿಯಾ ಆಗಿತ್ತು. ಅಂದ್ರೆ ನಮ್ಮ ಇಡೀ ದೇಶದಲ್ಲಿ ನಡೆಯುವಂತಹ ವೈಜ್ಞಾನಿಕ ಸಂಶೋಧನೆಗಳ ಬಗ್ಗೆ ಸರ್ಕಾರಕ್ಕೆ ಸಲಹೆ ಕೊಡುವಂತಹ ಹಿರಿಯ ಅಧಿಕಾರಿಗಳಾಗಿದ್ದಾರೆ. ಆದರೂ ಅವರನ್ನ ಇಲ್ಲಿಗೆ ಬನ್ನಿ ಅಂತಂದಾಗ ಬಹಳ ಖುಷಿಯಿಂದ ಬಂದರು. 2 ವಿಷಯ. ವಿಜ್ಞಾನ ಸಂವಹನೆ ಬಗ್ಗೆ ಪ್ರೊಫೆಸರ್ ವಿಜಯರಾಘವನ್ಗೆ ಇರುವಂತಹ ಆಸಕ್ತಿ ಒಂದು. 2ನೇದು ಭಾರತೀಯ ಭಾಷೆಗಳಲ್ಲಿ ನಾವು ಅದನ್ನ ಹೆಚ್ಚೆಚ್ಚು ಮಾಡಬೇಕು ಅಂತ ಅನ್ನೋ ಕಾಳಜಿ. ಅವರು ಬಹುಶಃ ನಮ್ಮ ವಿಜ್ಞಾನ ಜಗತ್ತಿನಲ್ಲಿ ಡಿಪಾರ್ಟ್ಮೆಂಟ್ ಆಫ್ ಸೈನ್ಸ್ ಅಂಡ್ ಟೆಕ್ನಾಲಜಿಯ ಸೆಕ್ರೆಟರಿ ಆಗಿದ್ದ ಮತ್ತು ಡಿಪಾರ್ಟ್ಮೆಂಟ್ ಆಫ್ ಬಯೋಟೆಕ್ನಾಲಜಿ ಅನ್ನುವ ಇನ್ನೊಂದು ವಿಭಾಗ ಹಾಗೂ ಪ್ರಿನ್ಸಿಪಲ್ ಸೈಂಟಿಫಿಕ್ ಅಡ್ವೈಸರ್ ಆಗಿದ್ದು DSAR ನ ಸೆಕ್ರೆಟರಿ ಆಗಿದ್ದಂತಹ ಬಹುಶಃ ಕೆಲವು ವ್ಯಕ್ತಿಗಳಲ್ಲಿ ಪ್ರೊಫೆಸರ್ ವಿಜಯರಾಘವನ್ ಅವರು. ಅಂಥವ್ರನ್ನ ನಾವು ಇಲ್ಲಿ ಬನ್ನಿ ಅಂತಂದಾಗ ನಾನು ಹೇಳದೇ ಇದ್ದು ಒಂದು ಸಣ್ಣ ಜಾಗ ದೊಡ್ಡ ಇದೆಲ್ಲ ಅಂತಂದ್ರೂ ಕೂಡ ನನಗೆ ಬಹಳ ಖುಷಿ ಆಗುತ್ತೆ ಅಂತ ಹೇಳಿಕೊಂಡು ಇವತ್ತು ಎಲ್ರಿಗೂ ಅರ್ಥ ಆಗುವ ಹಾಗೆ ಬಹಳ ಸರಳವಾಗಿ ತುಂಬಾ ಚೆನ್ನಾಗಿ ಹೇಳ್ಕೊಟ್ಟಿದ್ದಾರೆ. ಅವರಿಗೆ ನಮ್ಮ ಕಡೆಯಿಂದ, ಕಲಾಸೂಚಿ ಕಡೆಯಿಂದ ಹಾಗೂ ಕುತೂಹಲಿ ಕಡೆಯಿಂದ, KSTA ಕರ್ನಾಟಕ ಸೈನ್ಸ್ ಅಂಡ್ ಟೆಕ್ನಾಲಜಿ ಅಕಾಡೆಮಿ ಕಡೆಯಿಂದ ಅನಂತ ಧನ್ಯವಾದಗಳನ್ನು ಹೇಳ್ತೇನೆ. ಬಹುಶಃ ಮೈಸೂರಿನಲ್ಲಿ ಈ ರೀತಿಯ ಕಾರ್ಯಕ್ರಮಗಳನ್ನು ಇನ್ನಷ್ಟು ಹಮ್ಮಿಕೊಳ್ಳೋದಕ್ಕೆ ನಿಮ್ಮ ಬೆಂಬಲ ನಮಗೆ ಬೇಕು. ಇಷ್ಟ ಕಲಾಸೂಚಿ ಮತ್ತು ಚಂದ್ರಶೇಖರ್ ಅವರು
Sachi sir. Bani sir.
Thank you, sir.
Thank you very much.